Makowicz. I'm a postdoc in Germany at the University of Konstanz with Axel Meyer. And I'm going to be talking about how we need to look at maternal and paternal effects together simultaneously. So that's going to eventually be the take-home message from my presentation today. And so what we see in a lot of the literature throughout the past decade or so is these constant studies that focus on maternal effects, okay? And it's very few studies that focus on paternal effects, and very, very few, if any, look at the effects simultaneously. So this is a huge gap in the literature right now, and I think it's a really, really important gap when we're trying to decipher what the maternal and paternal environments are doing to the behaviors, to the life history characteristics, to the genetics of the offspring that occur from those parental environments. The other problem that I see with a bunch of these studies is that we've got a really big conflicting factor of parental care. Okay? So a lot of the model systems include mice, rats, humans, and stickleback. Okay? So most of these have mothers that care a lot for the offspring, so we can't really decipher whether or not the offspring are feeding off the mother's care in that really sensitive period early in life, or if it's the genetic environment that the mother experienced prior to giving birth, so the prenatal environment. And then you have model systems like the stickleback, where it's the reverse. It's the father that cares for the offspring. So the father that's caring for the nest, and his parental care behavior in this early life period might be what's influencing the offspring when compared to the maternal's environment, which is yolking up the egg. So this could be a huge conflicting factor in the literature, and we need a really good model system that can simultaneously look at maternal and paternal care or effects while eliminating that parental care effect. And this was pointed out by Verona's lab in September of last year, that this is a huge gap in the literature right now. So they say that ignoring paternal effects in our maternal models leads to a bias in our conclusions. And this is what we're seeing in the literature today. So I am going to be addressing two main questions. So how do maternal and paternal effects differ? So you can think of maternal effects as females that are with the offspring, so either embryos yolking up the egg, pregnancies, those can be influencing the offspring from the mother's point of view, the father's point of view, because males constantly generate sperm. The sperm that's generated during a stressful period might be modified when compared to a non-stressful period. So there's two different ways in which mothers and fathers can influence the offspring. So I'm going to be looking at that. And the second question is, it's really what I'm going to be talking about today. So early in life period effects. So whether or not there's a sex ratio difference between stressed mothers, stressed fathers, or together. Whether or not these stressed parents influence growth rates or sexual maturity of these offspring. And I was going to have a genetic variation, but I was very optimistic when I submitted my talk, so I actually don't have my genetic data right now, but we can talk later if you want to discuss that further. So I'll just be focusing on the early life history aspects of maternal and paternal stress. So I am using the Poeciliid fishes for a model system, and these are an excellent model system for this because they are live-bearing. The females have a follicular placenta. It acts very similar to the mammalian placenta, so you do have exchange between the mother and the offspring. Some species, this exchange from the mother to the offspring is very similar to what you see in mammals. You have a large amount of nutrient given from the mother to the offspring, and the offspring actually grow oversized during the pregnancy. And then you've got other species where there's very, very little exchange between the mother and the offspring, but they still have a functioning placenta. You also have really, really short generation times. You could get a generation anywhere between 20 to 35 days. So you could get several generations occurring within a short period of time. So this allows for a lot of really good life history, common garden type studies. They're very manipulative. So you can put several individuals into a tank, modify their environment, and then they will respond most of the time. But the best part is they completely lack parental care. So beyond the yolking of the egg or the pregnancy, there's nothing that the parents do to promote the offspring's growth, their health, or anything like that. Most of the times the mothers will actually consume the offspring if they're not removed from the tank. So no parental care in the system at all. So that will take away that really large confounding factor that we see in other model systems. So how do I do this? So what I do is I use a predator stress cube. So in the upper hand corner, you see this is 5 different tanks that have a predator stress cube. So if you can imagine it, all the water that's draining from this PVC pipe here is connected to a sump tank down here. That sump tank contains a predator, a Poeciliids, which is a natural predator of guppies. And so you've got the predator's pheromones, you've got conspecifics in that same sump tank, and then you've got alarm cues. So fish, when they're injured, they release these alarm cues, and it tells other fish to swim away really fast because there's a predator that's going to eat you. So we've got this water that's conditioned. So we've got a chemical stimulus. We also have this predator stimulus. Now, this picture, you can't really see it, and even in a good lighting system, you can't really see it. But the fish are right here, okay? So they're at the very top, they're at the very back of the tank, and they're in the structure, okay? So we know the chemical stimulus is working, because this is the anti-predator behavior for these fish. And as soon as you put this stimulus in here and you act like it's going to attack the parents, they scatter. You have them jumping and hitting the top of the tank. You have them trying to get away from the predator as fast. Whereas you don't see this, this behavior here where they're hiding really, really well in the control environments. They're actually coming up to the front of the tank expecting you to feed them. So we've got a lot of behavioral experiments that we did to confirm that these parents were indeed stressed. We also had, where the red label is a female treatment, so we have 10 individuals in here, and they're all female. Next to them we have 10 males in here, 10 females, 10 males, 10 females. So we have them separated by sex, so the females aren't experiencing sexual harassment stress, which is also a factor within the system. So what we did is, because we separated by sex, we could do this crossbreed treatment, to where we have a control treatment, and it's going to be from here on out CC, okay? Control male, control female. Then we have this CS, which is control male, stressed female. So the female originated from one of these things. Then we have a SC, which is a stressed male and a control female. So this one allows us to look at maternal effects. This one allows us to look at paternal effects. And then our final treatment is a stressed male, stressed female, so SS. This is where both parents are extremely stressed out. So they both originated from these tanks here. Okay, so we did a bunch of behavioral experiments that I could talk to you later about to confirm that the parents are indeed stressed. And then what we do is, after we do this crossbreeding treatment, the males are terminated, and then the females are raised in these same environments that they originated from. So this female would be in stressed environment, this female would have been in a controlled environment. That way you can get the prenatal stress that they should be experiencing. So any of that maternal exchange during the pregnancy is what we're really focusing on there. After the females have the babies will be transferred into a controlled control environment. So it's strictly the parent's environment and not the offspring's environment. So the offspring experience no predator at all. So what did we find? First, what's really, really interesting was we found a sex ratio bias. Okay? So to compare a predicted 50-50 expectation using a chi-square, this is significantly different in our stress treatments. Okay? And it was a 2-to-1 in a stress female only, almost 2-to-1 in both stress parents. So what's really interesting was when our male was stressed, when it was only our stressed male, we had almost a 4-time fold of female offspring. Okay? And I'll explain what I think might be going on here in just a bit, of where we can go with this type of research. It's kind of really interesting when you start thinking of the psychology part of it. What's really interesting, because a lot of the maternal effect studies that have occurred previously have looked at growth rate of offspring. However, when we look at our male offspring here, our female offspring here, let me orient with you real quick. So we've got birth of the individual, of the female, and then about 48 adults. So the darker blue, that's our PC, or our controls. Our lighter blue is our stressed female, but our control male. Our light pink is our stressed male and our control female. And then red is when both parents are freaking out, right? So they're really stressed. If we just look here at the females, we're not really seeing an effect of stress. But when we're looking at the males, we're seeing that stressed females are really what's influencing the growth rate of these individuals. When we look at male sexual maturity, so in guppies you have this gonopodium, which is a modified anal fin. This is used for intermittent organ. And then you also have sexual selection occurring, so you have a lot of color patterns. So gonopodial development that's occurring right here is you see that it's really the father, the stressed fathers, that are significantly different from the controlled treatments. And then that corresponds with the color development, where your controlled treatments are significantly different from all stressed parents. So male intermittent organ, gonopodium, is influenced by stressed fathers, whereas the color, which is a sexually selected trait, is influenced by both mothers and fathers. And these are extremely strongly correlated. So you can expect that as soon as the gonopodium is starting to develop, you will get about 10-day delay into color development. If you look here, this is where our control individuals are, and then all of our stressed individuals, or majority of all our stressed individuals, are here. So if you have a stressed parent, you're going to delay gonopodial development, and it's also going to simultaneously delay your color development. So to summarize, stressed parents both influence the male sex ratio. However, this seems to be that it's more so for stressed males, which might be because the males are the heterochromatic sex. So there might be some bias in X sperm versus Y sperm. That might be occurring there. So we know that stressed females influence the growth rates of sons, and that both parents had negative effects on daughters and their growth rates. And then I'm just going to skip to that, that's safe. But this is a take-home message, because we know already that the literature says parental stress has a major effect. However, we need to look at both parents. They're extremely different from one another. So with that, I'll